All living things, including you, started out as one singular cell. But we all know that you are not made of one cell. You are made of close to 1 trillion cells. This podcast is going to help us understand how living things get from one cell to many cells. So we're going to be talking about the cell cycle. In order for cells to grow and divide, they need to go through the cell cycle. Most of the cell cycle is just the life of the cell, a cell growing and doing its functions. However, when a cell needs to divide, it will go through the process of cell division to create 2 identical cells. The cell cycle allows for 3 really important things. Number 1, it allows living organisms to grow and get bigger so that we can go from one cell to many cells. It also helps us to repair old or broken down cells in our body. And finally, it provides continuity of life because when one cell divides into 2, those 2 cells are identical to each other. So let's break down the stages of the cell cycle. The first stage and the longest stage is called interphase. It is broken up into 3 shorter phases. The first phase of interphase is G1 or gap 1. This is the longest phase of the cell cycle because this is where cells are normal, doing their normal growth, and they're performing their normal functions. If they get the signal to divide, they will move into the S phase of interphase, which stands for synthesis, which means to create. During this time, the DNA will make a copy of itself during DNA replication. This is important because then each new cell that's created after cell division has a copy of the DNA. The last stage of interphase is called G2 or gap 2. This is where cells prepare for cell division. What happens here is that we create a bunch of extra organelles, cytoplasm and cell membrane, because we need to have enough stuff for each of the new cells that are created. And finally, that brings us to cell division. So when one cell divides to create 2 identical cells, 2 really important things need to happen. First, each cell needs to have a complete copy of the DNA. And each new cell needs to have organisms and organelles and cytoplasm. There are 2 different processes within the cell division cycle that allow the DNA and the organelles and the cytoplasm to be separated. The first is mitosis, which separates the copies of the DNA, and the second is cytokinesis, which allows the cell to divide. We're going to look at each of these stages independently. Before we do that, let's take a look at the major players involved in these processes. The first is the nucleus, which contains the DNA in the form of chromosomes. We're going to be taking a look at what the nucleus is doing throughout the process of cell division. The second are the centrioles. These are organelles that direct the movement of chromosomes during cell division, help keep everything nice and organized. So let's take a close look at cell division. Remember, cell division is made up of 2 parts. The first is mitosis, which is the division of the nucleus. The second is cytokinesis, which is the division of the cytoplasm. Together, they're going to allow us to create 2 identical cells. Mitosis is actually made up of 4 smaller phases. The first is prophase, then metaphase, anaphase, and finally telophase occurs right before cytokinesis. Let's take a closer look at what happens in each of these 4 phases of mitosis. The first is prophase. It's easy to remember because pro means first. So this is the first stage of mitosis. During prophase, our chromosomes are going to thicken into our X-shaped chromosomes. Then the nuclear membrane is going to dissolve, and finally our spindle fibers are going to form and attach themselves to our chromosomes. This is important because it allows our chromosomes to move throughout the cell. Let's take a closer look at our chromosome structure. Chromosomes are made out of DNA. The DNA molecule is very long, stringy, and extremely fragile. And if it were just to move around the cell during cell division, it could break and get damaged, which would be a problem because we wouldn't have that genetic information that cells so desperately need. So our DNA is going to actually wrap itself around proteins called histones. This protein complex is going to then continue to coil itself up into the X-shaped chromosomes that we can visibly see under a microscope. So this is a full chromosome. A chromosome, this X-shaped here, is made up of 2 sides. These 2 sides are called sister chromatids. One side is the original copy of the DNA, and the other side is the copy that was created during the S phase of interphase. They're held together in the center by something called a centromere. which is a collection of proteins that hold the two copies together until they're ready to split apart later in mitosis. This again helps keep the DNA protected and organized throughout the division process. So, once our chromosomes have thickened into our X shapes here in prophase, we move on to metaphase. Whenever I see the word metaphase, I think of middle. So during this phase, the chromosomes are going to line themselves up along the middle of the cell with the help of the centrioles and the spindle fibers. During anaphase, the spindle fibers pull the sister chromatids towards the opposite ends of the cell. Finally, during telophase, which is the end phase, also I like to think of it kind of like a telescope or a telephone, something that you could see or talk to someone that's far away. During telophase, the chromosomes are going to move towards opposite ends of the cell, far away from each other. And the nuclear membranes are going to begin to reform. By the end of mitosis, at the end of telophase, we should have one cell with two nuclei. However, this is not what we want. We want two identical cells, each with one nuclei. So that brings us to our final stage of cell division, which is cytokinesis. The term cytokinesis means cell movement, and so this is where the cell is actually going to divide into two. There we have it. The end result is two identical cells. Cytokinesis in animals and plants is slightly different. Cytokinesis in animals is going to allow for a cleavage furrow to form. A cleavage furrow forms, we can see it here under a microscope, because the cell membrane is fluid and very flexible. Okay, remember, animal cells do not have cell walls. However, cytokinesis in plants is much different because the plants have cell walls, and they do not have that flexible ability to form that cleavage furrow and pinch themselves into two. So what happens is in the middle of our cell, we start to form a cell plate. This is a cell wall forming in between the two newly created nuclei. The cell plate is going to continue to form until it reaches all the way through to divide our two cells. Here we can take a look at one underneath a microscope of a real cell. Those look like onion root tips. We can see the cell plate forming in between the two newly formed nuclei. Over time, those two cells will become two separate cells thanks to that cell plate. So, after cell division is complete with mitosis and cytokinesis, the cell will return itself to interphase, completing the cell cycle. Hopefully this was helpful, and stay tuned for the next podcast about the cell cycle control.